Hi everyone, my name is Jiani and I'm here with my partner Daniel. We'll be talking about the physiology of saltwater, Euryhaline, and freshwater elasmobranchs with you today. So just a little bit of an overview of our presentation. We're going to introduce you to elasmobranchs, what they are, and we're really going to dive into how salinity affects elasmobranchs. We're going to be specifically talking about stenohaline saltwater elasmobranchs, and then moving into Euryhaline and stenohaline freshwater elasmobranchs. We'll be covering a few model species that were particularly cool to us, like the bull sharks, the sawfish, and 2 different stingrays. We're going to finish off with the future of elasmobranchs as well as our conclusion. So first, we're going to introduce you to elasmobranchs. So they're a cool group of cartilaginous fishes that consists of sharks, skates, and rays. You can find them all the way back to 400 million years ago, and they're characterized by placoid scales and right dorsal fins. As of right now, there are about 1,000 living species, as you can see on the diagram on the right. So what's really cool about them is that they can actually survive in different salinities, as outlined in this pie graph here. So from the 1,000 elasmobranchs, about 95% of them are found in marine environments, meaning they live in saltwater full time. The other 5% regularly enter freshwater, and within the 5%, a tiny little sliver are elasmobranchs that only live in freshwater. So they don't enter saltwater at all, and they make up such a teeny tiny percentage of all elasmobranchs. So let's talk about some names, how we differentiate between these groups. So first we have the stenohaline saltwater elasmobranchs, and these are elasmobranchs that are only found in saltwater. In contrast, we have stenohaline freshwater elasmobranchs, and you guessed that they're only found in freshwater. The group that is in between the two are Euryhaline elasmobranchs, and these are the ones that can be found in either saltwater or freshwater. So first we'll be focusing on stenohaline saltwater elasmobranchs, and just a reminder, these guys are the ones that take up 95% of the 1,000 species. So this slide is to point out how much diversity there is. So here we have the great white shark, we have the manta ray, the Greenland shark, we have the little skate, we have the small spotted cat shark, and the small eye pygmy shark. And a fun fact is that the Greenland shark right here is actually the longest living vertebrate, so that's pretty cool. So you can imagine that cells in different environments with varying salinities, they will need regulation of bodily fluids to function properly. So in the diagram here, if they're in a hypoosmotic solution, then water is going to tend to move inside and salt is going to move outside. In contrast, if you're in a hyperosmotic solution, then water is going to move outside and salts are going to move inside. So the opposite. Stenohaline saltwater elasmobranchs are actually hypoionic osmoconformers. This means that they maintain a lower level of ions relative to their surrounding water, and osmotically, they'll conform to the salt water. What's really cool about them is that they utilize urea to stay isoosmotic with the surrounding water. And you can imagine how important urea is to these elasmobranchs because it's so expensive to make. It takes about 5 ATPs to make 1 urea. To conserve these urea, the efforts are made in the kidney, where 70 to 99% of the urea are reabsorbed, and diffusion could also occur across the gills, and that depends on how permeable the gill is. And this is outlined in this diagram here. So the bodily fluids and the medium are at really similar osmolarities, and in the kidney is where urea and TMAO are retained. The gills are going to try to block the loss of urea and TMAO by being less permeable. So even though urea sounds like a great deal, they actually have this adverse effect of disrupting protein function. So having high levels of TMAO can counteract the disruptive effects of urea. So these fish, they maintain a high level of TMAO and a high level of urea. And again, they're maintained by the kidney and they're blocked, their loss is blocked by the gills. Another thing that the stenohaline saltwater elasmobranchs have to worry about is salt regulation. So they have this really cool organ called the rectal gland, and it's an accessory organ that empties into the digestive tract, and they're able to excrete salt. What they found was that in marine elasmobranchs, rectal glands are actually found larger. So for example, in the cat shark, rectal glands are found larger in the cat shark compared to other elasmobranchs. And how the rectal gland works is that ions are moving in and out, and it's moving in and out through a combination of electrical and ionic gradients that move mainly sodium and chloride into the rectal gland epithelium cells.
So now we're going to move on to Euryhaline and stenohaline elasmobranchs, which live in freshwater. And so in freshwater, osmoregulation is a bit different than it is in saltwater. And so Euryhaline elasmobranchs retain lower concentrations of urea and TMAO compared to the saltwater elasmobranchs. And this is helpful for them to stabilize their internal osmolarity, especially when they're in situations where they can go between various salinities of water. It helps to just kind of keep it stable. And also in many freshwater elasmobranchs, the rectal gland is non-functional and is often more of a vestigial organ. Then moving on to our first model species, it's the bull shark. So bull sharks are Euryhaline elasmobranchs, and they live between the Tropic of Capricorn and the Tropic of Cancer, basically around the world along the coast. And they also move up coasts a little bit and inland as well. And they have been found in waterways around the world, even traveling over 1,000 miles up the Mississippi River or into the Brisbane River, as we see over here. And one thing about them, though, is that they're limited in how much they can do in freshwater because they have to return to saltwater for breeding. Following their acclimation to freshwater, they have been found to decrease rectal gland sodium potassium ATPase activity while increasing their kidney sodium potassium ATPase activity, which we see in this graph over here on the right, with the freshwater environments being in gray and the saltwater being in black. So we don't see any change in gill or intestine, but we do see an increase in kidney and a decrease in rectal gland. There's also been found to be a 350 milliosmol decrease in plasma osmolarity, which is correlated with that freshwater acclimation, which is quite significant. And we will see why that happens. And this is why. So bull sharks increase the expression of their kidney sodium potassium ATPases and their NKCCs when they're acclimated to fresh water. Yet there is no change seen in the expression of their urea transporters, which implicates that basically their urea levels aren't changing that much. It's more that their sodium and chloride levels are changing a lot. And that is why we see that 350 milliosmol difference in osmolarity. And as you can see here, all the urea transporters are kind of on this side, and they're still there in both cases, but we have the addition of these sodium potassium ATPases and the NCC up here, which is crucial for them. Now moving on to the large-toothed sawfish. It's another Euryhaline elasmobranch. However, it has a bit more of a restricted range, even more so than the bull sharks, in that they hug the coastline a lot more tightly. They don't venture out a lot, and they don't really travel upriver either. They tend to cling to the shorelines because they rely heavily on mangrove forests and seagrass meadows for spawning. And some of their populations actually live their entire lives in freshwater, such as the Lake Nicaragua population. And honestly, not too much is known about the large-toothed saltfish, simply because of their species status. Um, but what we do know is that when acclimated to freshwater, it has been seen that they have smaller rectal glands and they have lower levels of blood osmolytes than they do when they're acclimated to salt water. So they're likely to be very, very similar to the bull shark in many ways in terms of their osmotic control mechanisms.
So now moving on to the Atlantic stingray, another species that is Euryhaline. They are located on the border of the states and Mexico, so they are found in a much smaller region than the previous two fishes that we talked about. And what's really cool about them is that they actually live in habitats where saltwater mixes with freshwater, and they're also able to enter freshwater. They are actually one of the few species that are able to reproduce and complete a full life cycle in freshwater, and so they can tolerate a wide range of salinities, which make them a model species for studying Euryhaline elasmobranchs. One study that we looked at sodium potassium ATPase activity. So they found that the sodium potassium activity or ATPase are much more plastic, and this allows them to thrive in both freshwater and saltwater. So in the top graph here, you're seeing the differences in sodium potassium ATPase activity in the gills, and the bottom graph here you're seeing in the rectal gland. So in the gills, activity was found to increase with a decrease in salinity. So as you move towards the gill or towards the graph, you get a decrease in salinity, and this could be due to increased ion uptake in the gills. In the rectal gland, however, you see a little bit of the opposite effect. And you saw how sodium potassium activity is lower in freshwater, and this could be because they're more active in saltier water because they're working hard to excrete a bunch of ions. Another study we looked at reabsorption. So they were found to have a remarkable glomerular and tubular functional reserves in the kidneys. So what they did was that they had two groups of stingrays. One was kept at a control salinity and one was acclimated to a lower salinity for 24 hours. And they saw that the stingrays were able to absorb a lot of the sodium and chloride ions as well as urea in the lower salinity conditions than the rays were in the control salinity. And this is highlighted in the graph here where the black bars are the control salinity and the white bars are a stingray in a lower salinity. And you can see that the white bars are much higher. The same study also looked at two rates, the first one being the glomerular filtration rate and then the urine flow rate. And both of these rates were found to increase in the lower salinity group. The researchers weren't really sure about the mechanisms behind glomerular functional reserve, but they know that they do have a powerful functional reserve. And they also looked at urine flow rate, and again, it was found to be a little bit higher. And this makes sense because the stingrays are generating a large volume of solid-free water, which causes a higher urine flow rate. And this is highlighted in the graphs here. So in the top graph, we have the glomerular filtration rate, and it's much higher in this lower salinity in both hours, hour 1, hour 2, and the third one here, the group represents the average of the hour 1 and hour 2. On the bottom graph here, we're looking at urine flow rate, and they look pretty much identical to each other, meaning the lower salinity was found to be much higher.
And now we're moving on to our only denihaline freshwater representative, which is the Potamatrigonid rays. So there are 38 species of Potamatrigonid rays, with 36 of them unable to enter salt water because they have just adapted that much to the freshwater environment. And they live in the Amazon River and its many tributaries. And just to showcase the incredible diversity of these rays, here are just a few of them. And you can see like the wide variety of spotting patterns and the different tail lengths that they display, which is honestly incredible, especially given that they're such a conserved group. And so rectal glands, what rectal glands? They barely have them. The rectal glands of the pentametragonids are incredibly small, yet they are still present. As you can see for scale, this is a millimeter scale bar up here, and this is the rectal gland. This little sliver that is about 3 to 4 millimeters long and not even a millimeter wide. And so this implicates that they basically don't use their rectal glands at all, which makes sense given that they are in fresh water. Moving on to their urea levels, they seem to have very, very low levels of urea in their plasma. And this large decrease in plasma urea levels is likely due to the absence of the third and fourth loops of their nephrons, as those two loops are the location of the main urea transporters. And so we see on the bottom here a comparison of a saltwater array and a freshwater array, as described here. And you can see that on the left, you have the up and down of your typical nephron, culminating with, in these couple of loops back here, urea transporters just helping to reclaim that urea. Whereas here in your freshwater representative, you don't see that. You don't see the reclamation of urea by this mechanism, and so it just passes right on through. And so, at a glance, rectal gland sodium potassium ATPases exhibit lower activity levels in the bull shark, Atlantic stingray, and pentametragonids. And the same trend is likely to be observed in the large-toothed sawfish. However, it has not been investigated. The Atlantic ray increases its gill sodium potassium ATPase activity when acclimated to fresh water. And plasma osmolyte levels in fresh water vary depending on the species. So, for example, in the bull shark and the large-toothed sawfish, we don't see, or we see a decrease in pretty much everything, except in the bull shark, you see little change in urea. In the Atlantic stingray, you see all three of them actually increase. And in pentametragonids, you see little to no urea and very low levels of sodium and potassium. So now let's talk about the future of the elasmobranchs in fresh water. So here's a brief overview of the conservation status of some of the ones that we spoke about, as well as one that I find particularly interesting, which is glyphis. As a whole, they're not doing super well. The Atlantic stingray is of least concern, as well as two pentametragonid species. And those ones are definitely doing the best of all of them. The bull shark is the next up, which is near threatened, which is less than ideal. And then all the rest are doing pretty poorly. And this is largely because they are in danger. And so looking into the pentametragonids, they live in the Amazon, which is undergoing a lot of deforestation. And we're also seeing the adverse effects of this deforestation beyond simply the trees not being there, and that you have events like what happened last year in terms of the Amazon rainforest fires. And deforestation doesn't just affect them, it also affects the sawfish, because when you get the deforestation of these mangrove forests, which is actually very common for urban development, you lose that breeding ground for them and their population suffers as a result. Now, the glyphis sharks, as I mentioned briefly here, that I found interesting, there's actually a species of Euryhaline freshwater sharks, which is the only Euryhaline freshwater shark that is known. And it lives in the Ganga River in India, which is incredibly polluted. And as such, it is most likely trying to deal with the pollution levels, and it's probably struggling as well, as it is one of the critically endangered species that is actually very understudied to the point where we have no data on it, apart from just that it exists. And the final threat is sport fishing, which fortunately has been on the decline due to a lot of social awareness. However, it is still present. such as like in this group where you see these two teens and their whole family, just happy standing over this reproductive age sawfish that they just caught. Okay, so in conclusion, Stennahaline marine elasmobranchs maintain osmotic balance via the use of urea transporters in the kidney and by removing salts with their rectal gland. Euryhaline elasmobranchs tend to retain some urea while reducing their rectal gland and Ka function in order to remain neutral in freshwater. And Stennahaline freshwater elasmobranchs have almost completely eliminated urea retention and have a shrunken rectal gland. There's a lot less unknown about all these guys. However, it is challenging to study many of these species due to their conservation status. Hopefully, situations improve for all of them, and in the future, we'll be able to find out more about these incredible creatures. Thank you.
Thank you.